Hello everyone. Last week in lab we went ahead and we picked some yeast cells and we put them into a PCR reaction solution. Tubes look like these are the tubes that I did last week. And we did the PCR reaction. We ran agarose gel to verify that we got about a 1,000 base pair fragment produced, which should be basically the whole coding sequence for the FO13 gene. And the reason we're doing this is we're amplifying it so that we can go ahead and then submit this DNA in for DNA sequence analysis to verify the presence or absence of our mutation. These reactions should be pretty high frequency. Almost all the colonies should produce transformants that have the mutation, but in some cases they don't. So we have to verify that sequence so that we know what we're working with. This week to do the cleanup, because this solution here is got all the things in it, cofactors and things for Taq polymerase and all that sort of stuff. What we need to do is we need to purify the DNA out of these tubes, and to do that, we're going to use a very similar system that we did for plasmid prep, but it's just a little bit different. We have the famous collection tube. It's this tube right here, and a lot of different techniques use it. It fits in centrifuges really well, so it's nice. The cartridge, they call them GeneJet cartridge or something like that. So this looks a little bit different than what we did with plasmid prep. If you notice here, I've already labeled this one. It's already labeled number 2. And we'll be doing the purification using these. And in the supplies, you'll be getting 2 of these. We're going to just purify 2 of the colonies. And between the TAs and I, we determine which ones we think we should get you to purify. We don't, we're making a guess, because last week you did 4. This week we only want you to do 2, so we're going to do 2. So we'll have that. They'll have these. You have 2 GeneJet cartridges and 2 collection tubes. And you're going to have 3 solutions, one of which is this binding buffer, which is this kind of yellow color. And you're going to have a wash buffer, which is this clear colorless solution. It's got ethanol in it, among a couple of other things. And then we have an elution solution. And you'll have all these sitting on trays similar to this, sitting at your bench, ready for you to use. Here's the procedure we're going to follow today. What we need to do is we need to go ahead, and the first step is to add 40 microliters. We've already taken 10 out. 40 microliters of the binding buffer to the 2 samples we're going to purify. And I chose we're going to do samples 2 and 3. So I'm taking 40 microliters out, and I'm going to add that to this solution. So notice that it turned the solution yellow. And I will now, these are all independent samples, so please change your pipette tip. I'll take another 40 microliters out, and we'll add that to sample number 3. Notice I added it in, and I pipette up and down to mix it. So now we have 2 solutions that are yellow and 2 solutions that are red. The next step is we need to transfer that solution to the GeneJet cartridges. So I'll take the caps off. So we now have the cap off. So we have the cartridge here. This is number 2. We have the cartridge here. So we go ahead and we'll transfer the whole thing. So I need to set this to a larger volume. Since it's about 80 microliters, I will set it for about 85 microliters. I just want to be sure to transfer the whole solution out. Put the tip in here, and then go to tip toward almost to the bottom, all the way to the bottom of the tube, and just kind of suck it all the way up. And then I will Go ahead and take it. Now, whenever you're adding it in here, we need to put the tip in here, but do not touch that membrane on the bottom of the cartridge. So get it close to the bottom, but don't touch it. So we now have the solution in here. I'll put the cap on it. And for number 3, we'll go ahead and take that solution out. So you see that I have the solution in here. It's in the cartridge. And be sure to remember you need to keep this cartridge. So here we go ahead. The next step is to go ahead and centrifuge it. I won't show you the centrifugation steps on the video because you guys have already centrifuged a number of times in the past. You don't need to see that. I will be back in a minute because you need to centrifuge at maximum speed for 1 minute. I'll be back in a minute. I'm back, and here we have the wash solution at the bottom of the tube. I will go ahead and take the GeneJet cartridge out, empty out the collection tube for both of them. And now the protocol says to do the same, to wash the sample again, but this time with 200 microliters. So I'll change the pipette volume to 200. Get the wash solution here. Add 200 microliters of wash. And back off to the centrifuge. So I'm back now. I've now centrifuged the 200 microliter wash step. The protocol says to empty out the collection tube. Make sure, put it in here. Empty out the collection tube. And this wash solution contains a number of different solutions. It includes some ethanol. And in this case, the ethanol can potentially inhibit reactions. So what they recommend in this protocol is to go ahead and, now that we've emptied out, we've done 2 washes, a 400 microliter wash, a 200 microliter wash of this little membrane cartridge here. Now it says to go ahead and spin it again. And it's dry, just go ahead and force it through to make sure that you push through all of the ethanol to make sure the membrane is nice and dry. So we'll go back over to the centrifuge, and I'll be back in a minute. Okay, now we've gone ahead and we've centrifuged these washes a second time. And I have 2 conical tubes here. And what the next step is to do is to go ahead and transfer the GeneJet cartridges into conical tubes. Okay, so now we have that GeneJet cartridge in a microfuge tube, and I have it already labeled. I have these labeled 2 and 3. And the next step is to do the elution of DNA, and that's the final step. So we'll go ahead, and the elution volume is 25 microliters. So we'll go ahead and go down here, take the pipette down to 25 microliters. And we have the DNA elute solution. Added that to the cartridges. Caps back on. So here we have the GeneJet cartridges. You probably can't see the liquid in here, but there's 25 microliters of elution buffer right in here. So now back off to the centrifuge again. Another 1 minute spin at maximum speed, and I will be back. Okay, I'm back from the centrifuge. We've now eluted the DNA from the cartridge. If you look here, you can see there's a solution at the bottom of the tube, and the cartridge here looks basically dry. So we take this cartridge out, put it in the waste, close up the tube, take the cartridge back out, and the other one, close up that tube. Then I want you to submit these tubes. Make sure you have them labeled with your name, some kind of a naming convention, the sample numbers corresponding to the colonies you picked, and we need you to go ahead and put the mutation name on it. Like, for example, if it was glycine 93 to valine, put G93V on it. That way we know which ones are which, and we will go ahead and add the appropriate oligos to them and ship them off for DNA sequence analysis to verify the presence or absence of your mutation. Hopefully this was useful and helpful. We will see you later, and see you in the lab. Thanks.